Hello, I'm Dr. W. John Martin. This is the fourth of a series of new videos to remove any doubts regarding the existence of stealth-adapted viruses. The first video described the low stringency polymerase chain reaction, or PCR. The second, the culture of atypical viruses. The third video dipped into some of the politics, but it also highlighted the importance of having actual DNA or RNA sequence data to define a virus. Now this video will discuss the DNA sequence data that was initially obtained on the first cultured virus. Evidence that the patient had a virus infection was based on 10 solid observations. The patient had symptoms. Her blood tested positive for virus-related sequences using low stringency PCR. Cells from her blood cause a cytopathic or cell damaging effect when cultured with other cells. The cytopathic effect could be transferred to other cultures using either the cells or the fluid from a positive culture. The fluid was still infectious after being passed through a 0.22 micron filter that would remove any bacteria. The transfer did not occur, however, if the material from a positive culture was either boiled or treated with chloroform. The latter is consistent with the virus having a lipid envelope. As I'll show in a moment, virus particles could be readily seen in the infected cells. Now, over a period of 3 years, and collecting 18 blood samples and trying different techniques, positive virus cultures were obtained in 15 of the blood samples. In 6 of the positive cultures, the viruses were confirmed using electron microscopy. Now, the cultured viruses were also used in animal inoculation studies, which I will describe in a later video. Finally, the virus was submitted to the American type culture collection, which confirms the presence of the virus as it places such materials into repositories. Now this is an electron microscope photograph of a cell from a positive culture. It shows numerous virus particles in the nucleus, the cytoplasm, and especially within this larger vacuole. The particles measure approximately 180 nanometers in diameter, which is in the range of a herpes virus, and much larger than that of a simple retrovirus, which would measure about 90 nanometers. Now, if the positive polymerase chain reaction found in the patient is due to these viruses, then the virus culture should also be PCR positive. Indeed it was. This photograph is of an agarose gel of polymerase chain reaction formed on the patient's culture in comparison with the PCR performed on a control culture. The right two lanes, labeled M, are DNA size markers. It's very clear that the patient's culture yielded a very prominent band measured at approximately 1.5 kilobases or 1,500 nucleotides, and some smaller bands around 500 nucleotides. Now the larger band was excised from the gel and cloned into plasmids. Cloning and sequencing indicated there was actually 2 PCR products. One measured 1,484 nucleotides in length, and it was cloned in plasmid 1552. A slightly larger product measured 1,539 nucleotides, and it was cloned into plasmid 1554. Both sequences were submitted to the National Center of Biotechnology Information to be entered into what's called the GenBank. The sequences were also compared with all of the available sequences at the time in GenBank. The smaller product showed no relationship to any virus or cellular DNA sequence. The larger product, however, showed a statistically highly significant relationship to human cytomegalovirus. Certainly not identity. For example, over a region of 1,201 nucleotides, there was 58% identity with the nucleotides in human cytomegalovirus. This can be seen in this figure, which shows the comparison between the upper line in the different rows being the PCR product nucleotide sequence and the line just beneath it being the sequence of human cytomegalovirus. Each connection between the rows indicates an identical nucleotide. As one can see, it's close but clearly not the same as human cytomegalovirus. We could also conclude at this stage that the PCR assay intended to amplify retrovirus-related sequences actually cross-reacted with and amplified a cytomegalovirus-related sequence. These data were submitted to the American Journal of Pathology for publication in August of 1994. The next video will present additional DNA sequence on this virus and identify the species of origin of the cytomegalovirus. Thank you.